Hello all, welcome to Unit 3. Let me go back over here to our intro slide so we can see that. This is going to be the first video for Chapter 9. Now in Unit 3 we're going to be covering Chapters 9 and 10 and 11. So 9 and 10 are going to cover how we control microbes out in the environment and also within the body in artificial ways. And in Chapter 11, we're going to get into a lot more detail than we have so far about how microbes interact with the human body. And so some of that information will cover the normal microbes that live on and inside the human body, but then we're really going to spend a lot of time talking about infectious diseases caused by microbes. What is it about microbes that make us ill that's special and allows them to do so? And so that'll be the subject of Chapter 11. So we're really going to be shifting now into more medical microbiology type areas, which I imagine most of you guys will like, since most of you want to go into the healthcare industry in some way. All right, so for Chapter 9, we're going to be focusing in on the title of the chapter, Physical and Chemical Control of Microbes. And then Chapter 10 will include some of that as well, but more specifically we'll be looking at chemical drugs that we take to try to help us fight off infections. And so here in this chapter we're going to be thinking more along the lines of controlling microbes on instruments and surfaces. Now we will get into the skin and mucous membranes a little bit with antiseptics, but think more about environment and surfaces in this chapter, and then we'll get into drugs that go deeper into the body to help us fight off microbes when we get into Chapter 10. All right, so to start off with here in Chapter 9, let's start talking a little bit of terminology. A lot of this antimicrobial terminology gets thrown around and used and misused all the time, and a lot of these terms do have strict definitions that we should know. So there are several processes that are used to control microbes, and we'll go over the definitions for these. Sterilization. All right, so when something is sterile, that means, and we've covered this before, there are no detectable microbes, and that includes viruses, and it also includes, and this is really important, the bacterial endospores. Remember we talked a lot about those back in Chapter 3 and how difficult they can be to kill or inactivate. So in order for something to be sterilized, you have to remove all detectable microbes, and that includes endospores, which are generally one of the hardest things to get rid of, if not the hardest in a lot of instances. All right, so then another term, disinfection. So we hear this term used all the time. Now when you disinfect something, that doesn't mean you're killing and activating every single microorganism, but what you are doing is you're using some kind of chemical or a physical agent. Now a physical agent could be something like heat, and then we'll talk about some others as well. And you're trying to inhibit or destroy as many microorganisms as possible. You're especially focusing in on pathogens, the ones that can make us sick. And also you're trying to remove microbial toxins from things that you are disinfecting. And we'll talk a lot more about toxins. So these are chemicals that microbes produce that actually damage the body, and we'll talk a lot about the role that those play in causing disease when we get into Chapter 11. All right, so those are two different processes. Now, how often do you hear somebody say, I need to sterilize my kitchen counter? So you spray some 409 or something on the kitchen counter and wipe it up. Okay, I sterilized the kitchen counter. No, you are not sterilizing that surface. Or what if you go to the doctor and they start rubbing iodine on your skin? We need to sterilize the skin. No, that you are not sterilizing the skin in that instance. The only time sterilization occurs is if you have removed every single microorganism, and those types of things are not actually achieving sterility. So that's something important to keep in mind. Another term, disinfectant, we hear that all the time. Antiseptic, we hear that term all the time. And so these refer to chemicals, chemical disinfectants, chemical antiseptics that are used to control microorganisms. Disinfectants are used on non-living surfaces and tissues. Antiseptics are used on living tissues. So generally your antiseptics are going to be quite a bit milder than a disinfectant would be. A couple of other process terms: decontamination or sanitization. That's a cleaning and scrubbing process that's used to reduce the number of microbes down to safe levels. Often used with the food industry, but you can decontaminate other types of things as well. And that's kind of overlap with disinfection. You can decontaminate and disinfect at the same time. Antisepsis or degermation or degerming is kind of the same thing, but it's applying to living tissue. So you're scrubbing, you're washing to try to remove microorganisms off of surfaces, and those processes can be combined with antiseptics as well to help in those processes. All right, I like this figure from your textbook. This is an overview of the microbial control methods that we'll talk about here in Chapter 9. So this is kind of show you where we will be headed. So we're going to talk about physical agents. So that's going to be things like heat and radiation. Those are two key ones for removing, killing microorganisms. Then we'll talk about many different kinds of chemicals that help us achieve that. And then there are some mechanical methods as well, namely filtration, that we'll discuss. But this is kind of a good summary diagram to come back to at the end of Chapter 9 to help you review all of the different things that we have covered in here. And they've got things highlighted well. They're showing you which things are involved in disinfection and sterilization, antisepsis, and decontamination. And some other terminology as well on here. Germicides, bactericides, fungicides. Whenever you see that side on the end, hopefully that's reminding you of something like homicide, which is to kill a person. Germicides are things that kill microbes. So the word germ there is being used as a substitute for microbe. A bactericide, more specifically, is going to be a substance that kills bacteria. Fungicide will kill fungi. So some things are germicides. That means they apply to lots of different microbes, and some are more specific. And then now there are agents or processes that can be germostatic, bacteriostatic, well, no, that should say fungistatic instead of fungicidal. I just noticed that error there. And if something is static, it means that it's not necessarily killing the microorganisms, but it is basically putting them into, limiting their growth, putting them into a state of suspended animation, preventing any further growth. So just keep in mind that that's the difference between the side terms and the static terms as we go through this chapter. All right, this is a really good table that you should be, or figure that you should be familiar with as you move through this chapter. And what this is showing is that not all microorganisms are alike in terms of their resistance to various control methods. So this is just a kind of an overview chart showing you the types of microbes that are less resistant to control methods versus the ones up here at the top that are more resistant. So these are the bad guys, hard to control up here. These are the wimpier ones down here. Now just keep in mind, microorganisms are very diverse, so not everything will follow this this little chart. This is just kind of a guideline on here. But interestingly, so we just covered viruses in Chapter 5, and your enveloped viruses tend to be the most wimpy microorganisms. In general, they're the easiest to control, and those envelopes that they have on their surface tend to be pretty easy. It's pretty easy to basically melt or dissolve those envelopes. And then remember, your envelope viruses have those spikes in their surfaces, and if those spikes fall off, then those envelope viruses have no way to attach to their host cell. So in general, they're pretty easy to control, which is good, because if you think about like HIV, for example, as an enveloped virus, it's actually fairly straightforward to control HIV. And think about if it wasn't quite so wimpy. Think about if HIV could persist out on surfaces and so forth for days or weeks, and people could come along and pick up HIV off of a countertop or a toilet seat, things like that. Fortunately, that doesn't really happen for us. Now up here at the top, you're really the hardest microorganisms to control: prions, those infectious proteins that are pretty bizarre. We talked about those at the end of Chapter 5, and you're not likely to encounter prions, in your career very often, maybe not ever. But just keep in mind, if you do, they're very difficult to control, so you've got to be very careful. They're resistant. Bacterial endospores, we already talked about how resistant those are. The mycobacteria, remember they have the mycolic acid cell walls that are difficult to penetrate, so it makes those organisms difficult to control as well. Staphylococcus and Pseudomonas, each of those is a genus of bacteria that are medically important. They tend to be pretty difficult to control. Protozoan cysts, the cysts are a dormant form of protozoans. Those are hard to control. And as you move on down the line, you've got your gram-negative bacteria, fungi and fungal spores, non-enveloped viruses, most gram-positive bacteria, not all. But so you've got a spectrum here of microbes from less resistant to more resistant. I would like you guys, though, to know the ones up here at the top, the groups that are the most difficult to control, because that tends to come up fairly often. And you'll see things like this listed on the labels for disinfectants and antiseptics and things like that. They'll have listed as a high level of activity against enveloped viruses, or this is sporicidal. That's another term you guys should be familiar with. A sporicide is something that is strong enough to actually kill bacterial endospores, and so therefore most things that are sporicides are capable of sterilizing as well. If they'll kill the endospores, they're probably going to kill just about anything. All right, so some information about choosing control methods. How do you choose an appropriate control method to use? And first thing you have to ask is whether the instrument or whatever it is that you're using, the device that you're going to use with the patient, does it have to be sterilized, or could disinfection apply? And that gets into the devices, the equipment that you're using. Are those critical, semi-critical, or non-critical? Critical medical devices are going to be anything that will penetrate the skin or a mucous membrane lining inside the body. So keep in mind what body systems on the inside are lined with mucous membranes: your respiratory, digestive, urinary, and reproductive. So anytime you go deep to those areas, you must use, those are called critical medical devices, and those instruments have to be sterile before they can be used. Now, if you've got a device that's only going to contact a mucous membrane inside the body, so it's not going to penetrate that membrane, those can be considered semi-critical devices, and they need a high level of disinfection. Preferably, you're going to sterilize those types of things, but that may not always be possible or required. Then you got your non-critical devices. So if something's only going to contact the skin, it's not going to penetrate the skin, or things like furniture in hospital rooms and so forth, only low-level disinfection may be required in those cases. Is an item reusable? That's important. A lot of things that are used in medical settings are not reusable. They're disposable. And so, but if they are reusable, you've got to know critical, semi-critical, or non-critical to know how to handle those things. Looks like my little figure here is covering up some of the text, and I just noticed that, is the item reusable? What is its composition? So some things can't be treated with every possible chemical, or we're going to see heat can be used to control microorganisms. Well, some things can't be heated. So those, some of that's just kind of common sense. Will the agent penetrate to a necessary level? That's important as well. Think about if you have biofilms present, you're going to need something that's going to help penetrate and break apart the biofilm. Cost and labor efficiency, and this is really key. Safety, how safe is something? There are lots of things out there that might control microbes, but they're dangerous to expose personnel to. All right, another concept with these antimicrobial agents that we'll be talking about in this chapter, they have characteristic death rates. So a death rate is basically how long is it taking to kill a particular percentage of the population. So, as an example, so basically kind of the take-home message here is just because you spray a disinfectant on a surface, or just because you put something into an autoclave to sterilize it, sterilization, disinfection, these things don't happen immediately. It's not like you spray some alcohol on on a group of bacteria on a surface and they immediately detonate and blow up and they're gone. There are certain exposure times that have to be used, and so you have to follow those. When exposure times for a certain process or chemical are given, you've got to be sure that you're following that to achieve the level of disinfection or sterilization that's needed. And that's what they're trying to show you here on this diagram. Like, suppose for some antimicrobial agent, 90% of those microbes die with every 1 minute of exposure. So if you're starting with, over here on your y-axis, 10 to the 9th power of those microbes, that's a billion. Then after 1 minute, 90% have died. So you're left with 10 to the 8th, which is 100 million. You'd be right there. And then after 2 minutes, you're going to knock down another 90%, so you'd be at 10 million. And then 1 minute later, 3 minutes in total, you're going to be down to the 10 to the 6th power. That's 1 million. So you're kind of stepping down in terms of the number of microbes that are there. Okay, this is a table from your book, which is kind of nice as well. And it's just kind of re-emphasizing which microorganisms are the hardiest and which ones are the wimpiest for the most part. But what they're really trying to show you is a comparison of how different control processes or agents affect bacterial endospores versus the vegetative forms. Vegetative bacteria are the active metabolizing cellular forms. And so they're trying to show you, you know, if you are using moist heat, 80 degrees Celsius will kill vegetative bacteria. You need 120 Celsius for endospores. And they're also showing you exposure times for sporicidal liquids. 2% glutaraldehyde is a type of sporicidal liquid. But you would need a 3-hour contact time to kill all the endospores. Meanwhile, the wimpy old vegetative bacteria may all be dead within 10 minutes. So they're just trying to emphasize for you, you know, when we say endospores are difficult to control, they're not kidding. They are difficult to control. All right, so what affects this, the action of an antimicrobial agent or the death rate? And again, some of this is going to be kind of common sense. The number of microorganisms that are present. If you got more, it's going to take longer to kill them all. What type? You know, we've been over the how resistant different groups of microbes are. Temperature and pH will influence things. So if you put a chemical disinfectant on the surface of a table or on an instrument, for example, in general, that disinfectant is going to work a little faster when the temperatures are warmer, and it's going to be slower when temperatures are cooler, because those chemical disinfectants are killing microorganisms through chemical reactions that they're carrying out. And so chemical reactions tend to proceed more quickly in warmer temperatures, and they slow down in cooler temperatures. pH can influence how well these control methods or agents are working as well. The dosage, the intensity, you know, are you using an appropriate concentration? A lot of chemical disinfectants, you've got to dilute them with water to a certain percentage, and that's critical, because if you make a mistake in diluting it and you make 10% ethanol instead of 70% ethanol, that 10% ethanol is not going to do as good of a job at killing the microorganisms. The mode of action, so some control agents, their modes of action. A mode of action, you'll hear that in chapter 9 and 10, is how does the agent that you're using, now it could be heat, it could be a chemical disinfectant like some kind of alcohol, or in chapter 10 we'll be talking about drugs like penicillin that you take. The mode of action is how does it actually kill or inhibit the microorganism? Is it damaging the cell membrane? Is it denaturing the proteins? Things like that. That's your mode of action. Are other substances present? So if you're trying to kill microorganisms and they're present in a bunch of blood or feces or a solvent, some sort of, you know, other liquid that's present there, that can affect how quickly a control method will work. And again, some of those things are just kind of common sense. All right, so how do different antimicrobial agents, either physical or chemical, how do they control microbial growth? And so you want to check out this table from your textbook, because it shows the typical cellular targets. It could be the cell wall, the cell membrane, some aspect of metabolism, synthesis that goes on inside the cells. And then proteins are very often the targets of these things, because if you screw up the proteins of a cell, if those get denatured, proteins are your chemical workhorses. If a cell's proteins are not working, the cell is going to die. Cell membrane, if you can disrupt the cell membrane well enough, a cell's not going to be able to survive. The cell wall, we've talked a little bit about that before, like bacteria have the peptidoglycan cell wall, and if that gets disrupted in some way, then that cell's going to take up too much water and it's going to burst due to osmosis. All right, so that's just kind of an introduction to the physical and chemical control methods that we'll be talking about here in Chapter 9. Next video clip, we'll start getting into some more specifics about physical control methods that are used. You know, when are they appropriate, how do they work, etc.